Welcome back, everybody. We're diving into liquid chromatography
Yeah,
today.
fun.
We've got a whole stack of articles and blog posts about it. So
Yeah.
our mission, or should we choose to accept it, is to figure out how it works,
Mm-hmm.
why people use it,
Right.
and just all the cool stuff that it can do.
Definitely. A lot of people don't realize how often they probably encounter liquid chromatography in their daily lives.
Exactly. So to start us off, can you give us like the LC for dummies version of what it is? Like, what is the super basic explanation?
Well, you can think about it like this.
Okay.
It's basically a way to take a mixture and separate out all the individual parts of it.
Okay.
So it's kind of like if you had a big bag of M&Ms and you wanted to sort them all out by color.
Oh, that's good. Yeah.
It's kind of like that, except on a much, much smaller scale with molecules.
So instead of like sorting by color, are we sorting by size? Or like what are we actually using to sort these molecules?
We use their chemical properties to do the sorting.
Okay, but like practically speaking, how do we actually separate them? It's not like we have tiny tweezers, right?
Right, of course not. It's a little more complicated than that, but it all boils down to this concept called partitioning. Basically, different substances have different preferences for the environment they want to be in.
Okay.
Like, you know how oil and water don't mix?
Yeah.
They separate from each other because they have different affinities for different phases.
So how does that apply to LC?
Well, in LC, we have two main phases.
Okay.
There's the stationary phase, which is like a solid material, often packed into a column.
So like a filter?
You could think of it that way.
Yeah.
And the mixture flows through the stationary phase.
Uh-huh.
Now, here's the key. The stationary phase is designed to interact differently with each component in the mixture. Some components will stick to it more strongly, while others will just kind of breeze through it.
So it's like an obstacle course for the molecules.
Exactly.
Yeah.
And then there's the mobile phase, which is like a liquid that carries the mixture through the stationary phase.
So it's kind of like sweeping them along.
Exactly. So you've got this combination of the stationary phase acting as the obstacle course and the mobile phase carrying the mixture through it.
Okay, got it. And by the time they get to the end, all the different types of molecules are separated out, right?
Exactly. They come out at different times based on how they interacted with the stationary phase.
That's so cool. It's like a molecular sorting machine.
Yeah.
But how do we actually control this whole process?
That's where these specialized instruments come in, and they give us the precision and control that we need to make it all work.
Okay, so walk me through these instruments. What are we talking about here?
Well, first you have the pump, which is like the heart of the system. It pushes the mobile phase through the column at a very precise rate.
Gotcha. So it keeps everything moving along.
Yep, exactly. And then you have the injector, which introduces the sample into the mobile phase. It sounds simple, but it needs to deliver a very precise volume of the sample without messing up the flow.
Oh, so it has to be super precise.
Yes, definitely.
What else?
Then, of course, you have the column itself, which houses the stationary phase, our obstacle course. And choosing the right column is super important.
Oh, are there different types of columns?
Oh, yeah, lots of them. Different columns are designed for separating different types of molecules.
Huh. Interesting. Okay, so we've got the pump, the injector, the column. What else is there?
And finally, you have the detector.
The detector, right. And what does that do?
The detector is kind of like the eyes of the system. It measures the separated components as they come out of the column.
Oh, so it's like it sees what's coming out.
Exactly. And it gives us a signal that we can use to identify and quantify each component.
So it tells us not only what's there, but also how much of it is there.
Exactly.
Wow, that's awesome.
It's pretty amazing, right?
Yeah, it's like a whole orchestra of instruments working together to give us this detailed picture of what's in a mixture.
It really is. And the end result of all this is something called a chromatogram, which is basically a graph that shows us the separated components over time.
So it's like a fingerprint of the mixture.
Exactly. Each peak on the chromatogram represents a different component, and we can use that information to identify and quantify everything that's in there.
That's super cool. But this is all pretty technical. So like, how does this actually affect our lives?
That's a great question. And it might surprise you to learn just how many different ways LC is used in the real world.
Okay, I'm ready to be surprised. Hit me with some examples.
Well, one area where LC has a huge impact is food safety. Like, did you know that LC is used to test for pesticides in the fruits and vegetables that we eat?
Really?
Uh-huh. It helps make sure that the food we're consuming is safe and meets all the regulatory standards.
Wow, so LC is like a silent guardian protecting us from all sorts of nasty stuff.
You could say that. It's also used to analyze the composition of food
Yeah.
to make sure that it's what it says it is.
So it's quality control too.
Yeah. Like they use LC to make sure that olive oil is actually olive oil and not some other kind of oil.
Oh, that's interesting. I never would have thought of that.
Yeah.
So besides food, where else is LC used?
Well, another really important area is pharmaceuticals. LC is used throughout the entire drug development process.
Really?
Yep. From identifying potential drug candidates to testing the final product.
Okay.
LC is there every step of the way.
Wow. So it's like a crucial part of making sure that the medicines we take are safe and effective.
Exactly.
It's incredible to think about all the ways that this one technique is used to improve our lives.
It really is. And honestly, we've only just scratched the surface. There are so many more applications of LC that we could talk about.
Well, let's keep going then. I'm definitely curious to hear more. Okay, so we were talking about all the cool ways LC is used.
Yeah.
What other things can you tell me about? What are some of your favorite examples?
Okay, well, how about this?
Okay.
You remember a while back when there was that recall
Mm-hmm.
of that heart medication? Well, it turns out that LC actually played a key role
Really?
in detecting the impurity that caused the recall.
Oh, wow, that's crazy. So it was like a detective.
Yeah, pretty much. You see,
Yeah.
medications are pretty complex.
Right.
It's not just the active ingredient. There's all kinds of other stuff in there, like inactive ingredients, and sometimes even tiny amounts of leftover materials from the manufacturing process. Well, LC is so sensitive that it can detect even the tiniest amounts of these impurities.
Wow.
Yeah.
So even if there's just a little bit of something that's not supposed to be there,
Uh-huh.
LC can find it.
Yep. So in this case, with the heart medication,
Yeah.
there was this impurity that actually had a slightly different chemical structure
Hmm.
than the actual drug itself.
Yeah.
And even though it was present in super, super tiny amounts,
Uh-huh.
LC was able to pick it up.
Oh, wow, that's incredible. So they were able to see it on the chromatogram.
Exactly. Remember how we talked about how each peak on the chromatogram represents a different component?
Yeah.
Well, they saw this extra peak
Okay.
that wasn't supposed to be there.
Oh, so that was the impurity.
Yep. And because of that, they were able to identify the impurity, figure out that it was potentially harmful, and issue the recall.
Wow, so LC really saved the day there. That's amazing.
Yeah, LC is really good at distinguishing between very similar molecules.
Yeah.
That's why it's such a valuable tool for so many things, like
Yeah.
environmental monitoring and food analysis and forensics.
It's crazy how one technique can be used in so many different fields.
I know, it's pretty cool.
So you mentioned earlier that there are different types of liquid chromatography.
Oh, yeah.
Can you tell me a little more about that?
Absolutely. So it's kind of like this. Imagine you're trying to separate a bunch of different size beads.
Okay.
You wouldn't use the same sieve for all of them, right? You'd need sieves with different mesh sizes to effectively sort them out.
Yeah, that makes sense.
Well, it's the same with LC. There are different types of LC that are kind of like different sieves. They're designed to separate molecules based on specific characteristics.
Oh, okay, that makes sense. So what are some of these different sieves or types of LC?
Well, one of the most common types is called high-performance liquid chromatography, or HPLC,
Okay.
and it's known for its speed and efficiency.
So HPLC is like the express lane of chromatography.
Yeah, kinda. It uses higher pressure
Okay.
to push the mobile phase through the column, which makes the separation happen faster.
Oh, that's cool. So if HPLC is the express lane, what are some of the other lanes in the chromatography world?
Let's see. Well, there's one called size exclusion chromatography, or SEC, and it does pretty much what its name suggests. It separates molecules based on their size.
Yeah.
Larger molecules move through the column faster
Mm.
because they don't get caught up in the pores of the stationary phase, while the smaller molecules take a more winding path, so they take longer.
Oh, okay, that makes sense. So it's kind of like a maze where the bigger molecules can take shortcuts?
Yeah. Exactly. And this technique is really useful for analyzing large molecules like proteins or polymers.
Wow. So we've got HPLC for speed and SEC for separating by size. Are there any other types?
Absolutely. We've talked about separating by size, but what about separating by charge?
Oh, yeah, that's a good point.
Well, that's where ion chromatography, or IC, comes in.
I see, okay. And what's special about that one?
IC uses specialized stationary phases that are designed to attract and bind ions.
So it's all about those electrically charged molecules.
Exactly. It separates ions based on their charge and size.
Got it. And what kind of stuff is IC used for?
It's really useful in environmental analysis.
Okay.
Like, for example, IC can be used to measure the concentrations of different ions in water samples.
Oh, so it can help us assess water quality and stuff like that.
Exactly. And it's also used in pharmaceuticals to analyze drug formulation.
Oh, wow. So it's a pretty versatile technique.
It really is.
It's amazing to think that we started with this simple analogy of sorting M&Ms. And now we're talking about all these different types of LC and all these amazing applications.
I know. It's pretty cool how something that seems so simple can lead to such sophisticated techniques.
Absolutely. And you mentioned earlier that we've only scratched the surface.
Oh yeah, there's so much more to explore.
I'm all ears. What else is out there in the world of LC?
Well, as technology advances, we're seeing even more sophisticated and powerful LC techniques emerge.
Okay, now I'm really intrigued. What are some of these cutting-edge techniques, and how are they changing the game?
Well, one of the most exciting advancements in recent years is the development of ultra-high-performance liquid chromatography, or UHPLC.
UHPLC. That sounds pretty intense.
It is. It takes the principles of HPLC and kind of kicks them into overdrive.
Okay.
It uses even higher pressures and smaller particles in the stationary phase.
So it's HPLC, but even faster and more powerful.
Exactly. It offers way better resolution, so we can separate even more closely related molecules.
Wow, so it's like upgrading from a standard definition TV to a 4K Ultra HD TV.
That's a great way to put it. And it also achieves incredible sensitivity,
Okay.
meaning it can detect substances at super, super low concentrations.
That's amazing. So UHPLC is like the next generation of LC.
Exactly. It opens up a whole new world of possibilities for analysis.
I can imagine. But with all this talk about high performance and advanced technology, makes you wonder, are there any downsides to LC?
You're right. No technique is perfect. One of the challenges with LC is something called matrix effects.
Matrix effects. What are those? It sounds kind of sci-fi.
It's not as complicated as it sounds. It basically refers to the influence of other components in the sample, what we call the matrix, on the thing we're trying to measure.
Okay, so it's like if you're trying to find a specific book in a really messy and cluttered library,
Huh.
all the other books, the matrix can make it harder to find what you're looking for.
That's a great analogy. And in LC, these matrix effects can sometimes interfere with the separation or detection of the analyte.
Oh, so it can mess with the results.
Yeah, it can sometimes lead to inaccurate results if we're not careful.
So how do scientists deal with these matrix effects? Is there a way to clean up the library, so to speak?
Well, there are all sorts of sample preparation techniques that we can use to minimize the interference from the matrix. For example, we might use filtration or extraction or special cartridges to clean up the sample before we inject it into the LC system.
Oh, so it's all about prepping the sample properly to get the best results. What other challenges do you have to deal with when using LC?
Well, developing a really good LC method can be pretty challenging.
Okay.
It's kind of like finding the perfect recipe. You have to figure out the right ingredients, the right proportions,
Right.
and the right conditions to make it all work the way you want it to.
So it's not as simple as just plugging in the machine and pressing a button.
Definitely not. It takes a lot of knowledge and experimentation to get it just right.
So what are some of the things that scientists need to consider when developing an LC method?
Well, one of the most important things is choosing the right stationary phase.
Right.
We need to select a material that will interact differently with the components we're trying to separate.
Right, like going back to that obstacle course analogy.
Exactly. And then the composition of the mobile phase is super important too. We need to find the right mix of solvents to carry the sample through the column and help with the separation.
So it's like creating a spiral cocktail of solvents for each analysis.
Yeah, kind of. And then there are things like the flow rate, the temperature, and the injection volume. All those things have to be just right.
Wow, it sounds like a lot to keep track of.
It is. But when you get it right, LC is such a powerful tool.
Okay, now I'm really starting to understand how much goes into LC analysis. But with all these advancements in specialized techniques, I can't help but wonder, where is LC headed next? What's on the horizon for this technology?
Oh, that's a great question. And the future of LC is actually really exciting. There's a ton of research and development happening right now, pushing the boundaries of what's possible with this technique.
Well, don't leave us hanging. Give us a glimpse into the future. What are some of the big trends you're seeing? Well, don't leave us hanging. Give us a glimpse into the future. What are some of the big trends you're seeing?
One thing I think is really cool is the miniaturization of LC systems.
Oh, okay.
Like researchers are working on making these portable LC devices
Really?
that you can use for on-site analysis.
Wow, so you could like test water quality right at the source?
Exactly.
Yeah.
Or like check for contaminants in food right there in the field.
That would be amazing. It's like having a pocket-sized lab.
I know, right? It's pretty awesome.
What else? What other futuristic LC stuff is on the horizon?
Well, a lot of people are talking about automation.
Oh, interesting.
Like imagine robots handling on all the sample prep.
Okay.
injecting the samples into the LC system,
I don't.
and even analyzing the data.
Wow. So it's like taking the human error out of the equation
Yeah.
and making the whole process much faster and more efficient.
Exactly.
That's incredible. And what about combining LC with other techniques? Like we talked about mass spectrometry a bit earlier.
Right. That's another really big trend. Integrating LC with other techniques like mass spec
Yeah.
to get even more information about the samples.
So LC separates the molecules,
Mm-hmm.
and then mass spec gives us like the detailed ID on each one.
Yeah, it's like LC finds the puzzle pieces,
Mhm.
and then mass spec helps us figure out exactly what each piece is.
That's a great way to think about it.
And this combination is already having a huge impact
Yeah.
in areas like proteomics and metabolomics.
Wow. Those are some pretty complex fields.
They are, but that's what makes it so exciting.
Yeah. It's
Yeah.
like we're uncovering all these hidden secrets about the world around us.
Absolutely. And LC is giving us the tools to do it. I mean, even with all the technical stuff.
Yeah.
At the end of the day, LC is all about discovery.
Right.
It's about using this technique to learn more about the world and answer important questions.
Exactly. It's not just about the molecules themselves.
Right.
It's about what we can learn from them and how we can use that knowledge to make a difference.
Yeah, like in healthcare,
Uh-huh.
environmental science, food safety, forensics.
It's pretty amazing to think about
I know, right?
just how much one technique can impact so many different fields.
It's incredible. And honestly, we're just getting started
Right.
with new advancements and technologies being developed all the time. Who knows what the future holds for LC?
That's what's so exciting about it.
Yeah.
Well, this has been a fascinating deep dive into the world of liquid chromatography. I feel like I've learned so much.
Me too. It's been fun talking about all this stuff.
Absolutely. So, to all you listeners out there, if you could use LC to analyze anything in the world,
Yeah.
what would it be?
Good question.
What mysteries would you solve?
What secrets would you uncover?
The possibilities are endless.
They really are. So keep those brains buzzing.
Exactly.
Uh,
And who knows, maybe one of you listening will be the one to make the next big breakthrough in the world of liquid chromatography.
Thanks for listening.
See you next time.